Alright, hello everyone, welcome back yet again. So, in targeted immunodetection, in a sense, it's all a special variation of what goes on with protein chips, albeit a bit different. So, in this type of methodology here, it's really simple. All you're doing is you're using capturing agents that are always antibodies in a sense. I wouldn't call them capturing agents because they're not stuck to a place and trying to capture something. Not always, that is. But it's a similar concept. So that's why we have the name immunodetection. You're always using antibodies. It works just like protein chips, but in a different device, and you'll see how things flow. So we'd usually use the stuff in pregnancy tests and whatnot, and later on I'll give you an example. We'll work our way through a real life example using pregnancy tests. So before we begin with how it works, I will just consider the what part done. Okay, so on to how it works. Basically, you're primarily relying on something called ELISA. So ELISA, which is shown in the image right here and the image right here, stands for enzyme-linked immunosorbent assay. And although this sounds complicated, it's really, really simple. Just hang in there. These would give you color when they detect what you want, which is why I think it's similar to protein chips. It's just all about something binding to something and then getting color. But how do they actually give you this color? Well, you literally have, 5%. Okay, well, you literally have an assay with a capturing agent, that's an antibody right here, and an antigen right here. Unlike protein chips, where the capturing agent, in a sense, would be on the assay itself, that's why it's correct to call it a capturing agent in that case. In this case, it's not. I'm just using the term very informally. You can know if the antigen is present and how much of it is there, and all that good stuff by labeling the interaction between the antibody and the antigen with color. Now we'll see how that works. The reason you're getting color is literally just because you have an enzyme attached to yet another antibody, only getting attached to a combination of the antibody-antigen complex. Once it gets attached to that combination of an antigen-antibody complex, only then is it going to convert a certain substrate. Could be, you got lots of options really, but we're not going to dig into that. It would convert that substrate into another substance that is colorful. So just a quick overview. You have an antigen on the assay, an antibody attaches to that antigen, and it could attach to any part of the assay really, but let's assume it attaches to the antigen. We'll go through that point again, we'll come to it later. And then third point, you have another antibody attaching to the antibody that attached to the antigen. And this antibody has been labeled with an enzyme, and that's just fancy terminology for saying that an enzyme is attached to it. And this enzyme, once it sees that the antibody that it's attached to has attached to another antibody, which attached to another antigen, then only will it convert the substrate into a colorful substance, and then only will you get color in that specific region to show you that the antigen or the analyte is present. In this case, we use the word antigen because it's attaching to an antibody, but really it's just another analyte. Okay, so if you remember, I mentioned that this antibody right here could attach anywhere. It could attach to this surface instead of attaching to the antigen. But why is it that if it attaches to this surface, why can't we just have this antibody, the yellow one, basically running through the reaction and giving us color regardless? Now the thing is, the enzyme that we're labeling this antibody with is very specific. It will only work if this antibody is attached to an antigen, another antigen antibody complex. It's not going to work if it's just this antibody attached to anything else. And that is why it's important to have the enzyme being attached to another antibody instead of the purple antibody itself. Now, as you can see, if you have like a bunch of types of ELISA, and I know this is extra for curious students, but I just think that over here it comes in important just so you can understand the specific point. If we had direct ELISA, like I just said a few seconds ago, then it wouldn't be very specific, because if this right here chose to attach here, for example, you would still get color. So indirect ELISA really works well, but the most specific one, the best one, would always be a sandwich ELISA, which is the one that's used in pregnancy tests and all that. So in sandwich ELISA, you actually have a capturing agent, because this right here is right on the surface. It's the one that's actually on the assay, and the antigen will attach to it. Once the antigen attached to it, you'd have 2 more antibodies attaching, and that's the only way you can get the substrate to convert to a colorful substance. So yeah, now that that's done with, I'm going to tick off the how part. Okay, now we know what this is and how it works. We need to look at clinical applications and results interpretations. So with clinical applications, like I said, let's look at pregnancy tests. This sort of stuff can be applied in so many different places. It could be seen in oncology, which is the study of cancer and whatnot. Toxicology, which is all about poisons, testing for autoimmune diseases, and even pregnancy tests and so on. I think it's important for you to get out of the oncology box for a bit. So I thought the pregnancy test would be a nice way of explaining this. So this is a small sketch of what a pregnancy test looks like. If we zoom in specifically into this part, we get this right here. Alright, so let's see how this works. You first have an analyte. In our case, it would be urine with the hormone HCG, and this would carry itself over the enzyme labeled antibody, right? Like we already know that this enzyme labeled antibody, once it attaches to the analyte, it's gonna go over and stuff is going to get attached to the primary antibody. So in our case, they're using sandwich ELISA. So this right here is your capture antibody. So the analyte labeled antibody complex is just going to go right over, it's going to attach to the sandwich, it's going to attach and give you like a sandwich complex, and that would be on the test line. It's not all going to be on the test line though. You're still going to have some going over, some excess going over and getting on the control line. And there's still even going to be some excess reagent, and that's just going to like wash off in a sense. So if the analyte has the substance that we're looking for, then we're going to get color on the test line. You're always going to get color on the control line anyway, because it's a control line. And that's why when we have a positive result, we have 2 different lines, in a sense, 2 different colorful lines, because the test line shows color if a lot of HCG hormone is present. I'm sorry for the background noise, it's not the quietest time of the day. And if the HCG hormone is not present in the amounts that we're looking for, then you're just going to get 1 line or a negative result. And yeah, that would be it really. we are done with clinical applications. Your interpretation of results would literally be those 2 lines or 1-line thing, so I would consider that done with too. Okay, so I'll see you in the last video, which is all about comprehensive proteomics.